Good afternoon, everybody, and welcome to the research session of this fantastic NODCC meeting. I'm Linda Richards. I'm a professor of neuroscience, and I'm actually also chair of a department at Washington University in St. Louis. I've been working on studying the Corpus Callosum for 27 years, and today, I wanted to talk to you a little bit about our motivation and goals in research and some of the current challenges that are in the field, as well as a little bit of a few updates. And then we'll have in this session some speakers. Lynn Paul will be speaking next. We will have a speaker online who is going to give an update on the genetic research that Elliot Scher and my group and Lynn are involved in. And then we will have a couple of speakers from Professor Warren Brown's lab and Dr. Lynn Paul's lab. Then we will have a couple of speakers who actually are looking at the lived experience of people with corpus callosum disorders. Marie Maxfield and Michael Shanahan, both from Australia. And then finally, we will have a Zoom talk from Peter Shakes, who is also from Australia. She'll be joining us by Zoom. All three of those people are currently undertaking their PhDs, and their topics are on corpus callosum disorders. So we're really excited to have them join us today. So I'm going to kick it off with some of the current challenges in disorders of the corpus callosum. So as you are probably very well aware, disorders of the corpus callosum are vastly variable in their range of structural, genetic, and cognitive phenotypes. I'm going to use this word phenotypes. It's a kind of scientific term. It just means outcome, really. The way the genetic phenotype might be the outcome of that genetic mutation. The cognitive phenotype means the behavioral symptoms, things like that. So you'll hear the word phenotype quite a bit. But as I said, one of the major challenges is that we're not talking about a simple single disorder here. We're talking about a range of conditions that all come under the umbrella of corpus callosum disorders. And so this makes the challenge of understanding and treating these disorders very difficult for the individual who is having the lived experience of disorder of corpus callosum, their families, their doctors who are looking to provide the best medical care, and scientists. So doctors, this is really also current state of affairs. Doctors can currently provide a structural diagnosis, but they don't really know what it will mean for you in many cases. So one of the things that our group of researchers does a lot is speaking with pediatric neurologists, clinical neuropsychologists, neurologists, neurosurgeons, many in the medical profession. We each give talks at their conferences. We give talks in their departments to try and educate them about corpus callosum disorders. There's a lot of misinformation in the medical community, and one of our goals is to try to address that. And so usually doctors don't know exactly how to treat your specific DCC phenotype. And they often now resort to therapies that work for other conditions. Autism would be one, maybe ADHD, drugs that work for ADHD. They may be actually treating ADHD within your child. But at the moment, that's the current state of affairs. And they're often not able to provide much guidance or prognosis prognosis is something that we are really working hard on in the research team. We would like to be able to provide families and doctors with information from the moment of diagnosis as to potential outcomes, potential phenotypic outcomes for that individual. And to get that kind of information, we have to do longitudinal studies, of course. We have to study people over a long period of time so we know this group of individuals have these kind of challenges and these kinds of things they're really good at. And this group has a different set. But we need to follow people over time to get that longitudinal data to provide that information for new families who get a diagnosis. Right. So today I wanted to focus on how can research improve the quality of life for children and adults with DCC. I think that that is the common goal that we all have. And I wanted to explain a few things about the research and the science translational pipeline and what we're driving towards. So I think I'm not sure if the lights can go down a little bit in the room. They cannot. Okay, so I hope that you can see the slides okay. What this shows is, I'm not sure if I can point. I don't know if you can see that. Is this a pointer? No. This one? Yes. Okay. So over here, it says idea. Well, yes, that's the kind of what does the corpus callosum do type of idea we're talking about. I'm going to talk a little bit here about basic research. And I just put this up because, of course, what we'd all eventually like to do is to get to a point in which we can run clinical trials of potential therapies for different symptoms of people with disorders of the corpus callosum. And what does that actually mean? What it means if we were to run a clinical trial, and let me just talk about that section first, is that these are the phases that are involved in running a clinical trial. You don't need too many people for a phase 1, which is really just understanding whether the treatment is going to have any adverse effects. To go into phase 2, you need a few more people. And as things progress, you need a lot more people. And then out the other side, hopefully you will have some treatment. It could be a drug, or it could be a behavioral treatment or something else, which has then evidence that that works. This is what we really want. We want to have evidence that a therapy that we're trying to provide actually works. And we get that evidence through running clinical trials. All right, now the science part of it, I wanted to talk a little bit about the basic research side here. So here are the scientific challenges at the moment. What does the corpus callosum do? Amazingly, and you have literally the experts in the world in this room today. There are some more experts that are in our research consortium that are in Europe and elsewhere in the world who we collaborate with. But believe it or not, we still don't have a good understanding of exactly what the corpus callosum does. So how does the corpus callosum underpin brain function and cognition? And you know the work of Professor Brown and Dr. Lynn Paul and their groups. They've been working on this for decades. And it's still, there's so much that we're uncovering all the time. So how the corpus callosum is important for brain function, and more importantly, what happens in a person with a corpus callosum disorder is what we're really trying to understand. And how much does this change throughout life? You're probably aware that Dr. Paul is doing a longitudinal study from the moment of birth through childhood trying to gather that longitudinal natural history data from parents, which is going to be crucially important for running any kind of clinical trial. How do disorders of the corpus callosum arise? I would say that's more in the realm of perhaps the work of Elliot Scher and myself, trying to look at what causes corpus callosum disorders. And I'm going to talk a little bit about that a little bit later. And how does the corpus callosum develop? Again, the kinds of questions that Elliot Scher and I are looking at. What genes are involved and how do these genes function? So later, we will be hearing from Manu Argili, who is a scientist working in Elliot Scher's lab. And we have just undertaken a massive genetic study, and she's going to be giving you the results of that. So I won't preempt that work. But of course, knowing the gene is the first step. We need to then understand how that gene actually functions. What does it do in corpus callosum development? We also, it's very important for us to understand what other conditions are associated with disorders of the corpus callosum and why. Why do some people have epilepsy? Other people's have cerebral palsy type motor symptoms. Other people have intellectual disability. You know, all these things that go along with disorders of the corpus callosum. What are all these conditions? Are they related to the corpus callosum disorder? Are they not related, but maybe it's the same gene doing different functions in different organs? It's very complex. So these questions all lie at the heart of the work that scientists are doing to understand fundamentally how the brain develops and how it functions. So I wanted to just make the point that this work is important for this community. It's important for the work of scientists working on development of the brain and how the brain functions. But it's also a higher calling than that, actually, believe it or not. We are trying to understand how the brain actually works. And this will have implications not only for corpus callosum disorders, but for the huge amount of different conditions that are associated with mental illnesses and neurological disorders of other kinds as well. This research trying to understand how the brain works through working with people that have a differently wired brain has the potential to have impact well outside of just the community of people with a corpus callosum disorder. So those of you who are involved in research, thank you, and know that it really can have impacts in the whole area of understanding the brain. Okay, so I wanted to just talk a little bit about genes because in the last 5 years or so, there has been a real rapid acceleration in our ability to diagnose conditions through genetics. And you are going to hear about the specific genes involved, but I wanted to talk a little bit about how that landscape is changing in terms of family organizations as well. So as you know, a disorder of the corpus callosum is a structural diagnosis with a scan, a CT scan, an MRI scan, or an ultrasound scan. The genetic understanding of these disorders of the corpus callosum is really rapidly gaining pace, yet still 50% of people with a DCC have no genetic diagnosis yet. We are working hard to try and provide that genetic diagnosis in the field. And I'm giving another talk later in the conference on genetics and DCC, and we can talk a little bit more in that talk about why we still haven't got 100% of people diagnosed with that have a genetic condition. So why is a genetic diagnosis important? Because the vast majority of DCCs are caused by a genetic alteration in a person's genome. We know that there are some environmental effects that can cause a corpus callosum disorder, but the vast majority are genetic. And it should be noted that a genetic diagnosis may be quite complex. So you may have a genetic diagnosis where it's a single gene, but actually there still could be other genes involved. Could be that gene is causing the majority of the phenotype, but there may be other genes involved. And it's good for you to know that. A genetic diagnosis is an important first step in finding biological treatments, such as drugs, gene therapies, etc. And a genetic diagnosis can also help you find other people with the same genetic condition. Everybody is, looking on the internet now and able to find the other people that have the same genetic condition. And so what I've put up here are just some websites of different family organizations that are now springing up around single gene conditions. Each one of these is associated with corpus callosum disorders. All the ones on this slide, and I won't go through what they all are. We can talk about them later if you like. But every one of these, the kids have a corpus callosum disorder. Whoops. Just go back. Right. Here are a couple more. The ones on the previous slide were all single gene family organizations. These are also single gene family organizations. And then there's family organizations around chromosomal abnormalities. Something that we are discovering is that chromosomal abnormalities associated with chromosome 8 are highly prevalent in corpus callosum disorders. And then there's a whole bunch of genes that are highly associated. Your gene might not be on here. This isn't an exhaustive list, but there's a lot of genes on here that are highly associated with corpus callosum disorders. And there's no family organization for these that I could find on the web. So what I did was take all the genes that are commonly associated with corpus callosum disorder and just do a Google search to see if I could find a family organization. So, all right. So what's the main goal of these family organizations? I'm sure many of you might even be in those family organizations. So I don't want to make presumptions, but from their websites, many of these family organizations, and remember, all the kids have a corpus callosum disorder. They often have something else as well, but they all have a corpus callosum disorder, except that they know their gene. They often form where the kids are very severely affected, and they have impacts from the gene that are outside the corpus callosum disorder. And the main goal, of course, is to find treatment or a cure and a way to repair that specific gene. Now, something else that's quite obvious from just surveying this is that all of these single gene conditions are rare disorders. So this means that there are fewer people with the disorder when you compare it with something like cardiovascular disease or breast cancer or heart disease. Even when the same gene is disrupted, different mutations can have different effects. So you might have the same gene in two different people, but a different mutation in different people can cause a different phenotype. That's how complex it is. And so what a lot of organizations, I'm going to talk about one in a moment called Global Genes, are suggesting or doing is trying to bring these family organizations together to kind of aggregate disorders that have a similar phenotype. And that way they can then run clinical trials, because you need a lot of people to run a clinical trial. So you can't run a clinical trial on a handful of patients with the disorder. So I wanted to talk a little bit about this organization called Global Genes. This is an organization that sprung up from one family organization with a rare disorder, not a corpus callosum disorder, but they have this, under this umbrella here, they have an organization called Global Advocacy Alliance, and that is a group of family organizations. It's an umbrella organization of family organizations. They also have a very interesting database called RareX that is across all these different family organizations. And that is a database that is owned by those family organizations. They put clinical information into that, and they've got it set up in a way that pharmaceutical companies want the data. So it is a way that they are making themselves clinical trial ready. And on this global advocacy website, there's some really great videos, which I highly recommend, on research and how to become research ready as an organization. And so what are they doing to support these research efforts? So there are sort of two ways to go about this. And I want to talk about each way. The first is to try and support basic research to understand what the gene actually does. And now I'm just talking about the gene side of it, and I'm going to come back to the behavioral side of it in a moment. But basic research, if like nothing is known, you've got a genetic diagnosis, but nothing very much is known about that gene, then you're going to need some basic research to figure out actually what is the gene, where is it expressed in the brain, and what does it do? You will need natural history data. These are what these family organizations are trying to promote. They might need cell lines, biologicals, so they can look at gene function and do drug screening. They might need mouse models where they've knocked out the gene, removed the gene, made the same mutation that we find in humans in an animal model to look at how it affects brain development. And they might be doing human cognitive research, the same kinds of things that our organization, the International Research Consortium, that Lynn is going to talk more about in the next talk, does. All right. The second is that they focus only on identifying treatments without knowing what the gene does. So you can do that. You can take a kind of broad approach where you can actually screen for known drugs or known small molecules and try to repurpose drugs that might have been used somewhere else. But where do you start? So for that, you would still need to use biologicals, human cell lines. And if anybody would like to discuss any of this later, I'm happy to talk about it. What does that mean? Human cell lines. We can make cell lines from a skin sample from an affected individual. It will still have the same gene mutation in it. And then we can make neurons from that skin sample. And then we can look at the neurons, and if they have a phenotype that's different to a neurotypical person where we've taken the skin cells, that gives us something, a change in the cell that then we can add drugs to and see if we can rescue that back to the control phenotype. Hopefully that makes sense. We can identify which symptoms need treatment. These are the family organizations. What are the most pressing symptoms that they would like to run a clinical trial for? And they can raise funds to run these clinical trials. Was it 1? 5, great, okay. All right, so just wanted to give you that information. I wanted to now turn away from the genetics and talk a little bit about treatments for the actual corpus callosum disorder. What about all the people that don't have a genetic diagnosis? And so I just wanted to make a few points and we can discuss them more. So even though many genetic mutations can cause a disorder of the corpus callosum, the actual cognitive phenotype is really quite similar across those different genetic disorders. So here you have the power then to potentially be able to run clinical trials because you see that people do have similar cognitive outcomes. And I am not the expert on that. You have two experts sitting down the back that'll be able to answer questions about it too. Another point, though, is that the behavior and the cognitive phenotypes are really tough to treat. They are hard to treat because their behaviors, they impact mental health, cognition, not only in DCC, but in all conditions that affect the brain. So, you know, in the whole field of neuroscience, we are really we're making great strides in understanding the cause of Alzheimer's disease, for example, and there are actually treatments coming out for Alzheimer's disease. For mental illnesses, we are so far behind because we really still don't understand what causes depression, anxiety, schizophrenia, etc., autism. We still don't know exactly what causes these. So basic knowledge about the brain is needed to really accelerate those areas. So of course, why is the corpus callosum involved here? And that is because your brain does all these amazing different functions, and it can do all these amazing different functions because it is wired up as in circuitry that actually encodes the information that enables your brain to do all these things. And the corpus callosum is the largest fiber tract connecting the two hemispheres. So we are talking a mega uber circuit when we're talking about the corpus callosum. We are very keen to understand how disorders of the corpus callosum occur and how they affect people. And I put the AusDoc group up there. I've been working with the AusDoc group for a very long time. And here I am super excited to work with the National Organization for Disorders of the Corpus Callosum. In our group, we work on in three areas. Firstly, we work with people that have corpus callosum disorders. We then also look at mouse models of the genes that are disrupted in corpus callosum disorders. And we also work on a small little marsupial. And I can tell you a little bit more about why we work on a marsupial. Marsupials don't actually have a corpus callosum. They didn't evolve a corpus callosum. But the more interesting thing about these animals is that they're born at a very early stage of development into the pouch. So we can study mechanisms that occur before birth in this marsupial. And we have a colony of these marsupials at WashU. So our lab is interested in early brain developmental events that set up the wiring of the brain and then how that brain wiring underpins cognitive function. And I don't have to tell this audience that the corpus callosum connects the two brain hemispheres, but perhaps it's worth pointing out that people with corpus callosum disorders actually have very interesting rewired brains. So it's not just that you take the corpus callosum out and it's gone. Actually, the brain during development is very plastic and it rewires itself. And so we're trying to understand, how does that rewiring impact cognitive function? Okay. We know a lot about how the corpus callosum develops from mouse work. This is a mouse brain, and it shows you the different stages this is the cortex of the mouse. The cells for the corpus callosum are born in the cortex, and they project their fiber towards the midline, they cross, and then they find their correct target in the contralateral hemisphere, and then they innervate there. We can use mice to study these things. And previously, work from my group and together with Elliot Scher actually described the cellular processes, and we're uncovering more about the molecular processes that go into forming this midline bridge that's absolutely required for the corpus callosum to form. We also know a lot about genes that are involved in this. This is just one of those, deleted in colorectal cancer. These people have variations on partial agenesis of the corpus callosum or complete. These are all the gene mutations that are involved, just to give you a flavor of some of the advances that we are making. We also know from all the work from Lynn Paul, Warren Brown, and a little bit of work from our group that those with corpus callosum disorders demonstrate exaggerated vulnerabilities to social persuasion and are less aware of possibly being deceived. They're actually more gullible. So every brain contains unique information, and my message to you is, can we together understand how the brain works? So all of the research groups here have research that we're undertaking here at the NODCC conference. This is my team. They're sitting right outside, and we would love you and your families to be involved. So thank you. So Lynn is going to give the next talk. Would you like to ask any questions in the meantime while Lynn is setting up her talk? You might, there is a microphone there. Yes. Yeah. Yeah. Semi-lobar holoprosencephaly is not that common. And we can talk about your specific case. It's not that common. So that is, we would need to have a look at the scan exactly to be able to say, but it's a little bit different than the bridge perhaps mechanism that I was just talking about. Yeah, but thank you for that. Yeah, you go ahead. I'll just stand over here. Is it possible to turn the mics on? Yeah. Oh, perfect.
Thank you for a very interesting overview. It seems like there is considerable research going on looking at the differently expressed genes in relation to the disorder. My question is, in cases when phenotypes have not down or upregulated genes, but actually having the full deletion,
Yep.
are you aware of any research that trying to address this link?
Yep. Yeah, so the genetic research is addressing both what we call loss of function mutation. So that's where the proteins are made from genes. That's where the protein's not going to either be made or not going to function properly. And then there are also gain of function mutations. So that's where the protein actually has a hyper kind of phenotype. We're looking at all of those. So there's definitely, depends on the gene you're talking about, but.
Right, but those are dealing with the differently expressed genes. What
Yes.
about when genes are not there, when there is a full deletion? Is there any research that is addressing this kind of condition?
In fact, that is the case of how we usually make mouse models. There are some genes that are completely deleted. Are you talking about a chromosomal abnormality as a CNV?
I'm talking about like Midas syndrome.
Sorry?
Midas syndrome.
Midas syndrome. Elliot, do you want to make a comment? No.
I could
come and
go for
see
it.
us. Come and see us afterwards. But certainly, all of these things are being modeled in animals, and we're trying to figure out what the deletion would be doing. Yeah. Any other questions? Yeah. And we're trying to figure that out, but in the face of saying this in front of like the world's expert sitting down the back there, Dr. Brown, we have a theory. It's around something called metacognition, so deficits in metacognition. And that is sort of personal introspection and insight, and that there's a deficit in that, which means that it's much harder to know how you're doing in a social interaction, in a social context, or in an interaction with another individual. You need that internal feedback, and that's the hypothesis. Yeah. I'm going to hand it over now to Dr. Lynn Paul.